Assalamu alaikum. I'm here to tell you about the physiological basis of the neuronal function, and I'm starting this with the name of Allah, who is the most beneficial and the most merciful. Firstly, I will tell you about the resting membrane potential. A typical neuron at the rest is the more positive electrically outside than the inside the cell membrane, and this net difference in the charge between the inner and the outer surface on the non-conducting neuron is called the resting membrane. There are the three mechanisms of the resting membrane potential, that are the electrogenic ion pump, Dunning equilibrium, and the diffusional potential. These are the three basic mechanisms of the resting membrane potential. In this diagram, you can see the mechanism of the electrogenic ion pump. Basically, electrogenic ion pump is also known as the sodium-potassium ion pump, and this diagram will describe about the electrogenic ion pump perfectly. In this diagram, you can see that sodium potassium pump exchange the 3 sodium for the 2 potassium per ATP hydrolyzed. This mechanism of the electrogenic ion pump contribute the normal membrane potential. If we talk about the Dunning equilibrium, this mechanism contributes to small membrane potential because the intracellular fluid has a higher protein concentration than the extracellular fluid. The third important mechanism which contributes in the resting membrane potential, which is the existence of the marked diffusion potential for the various ions across the membrane, is known as the diffusion potential. And there are the three basic aspects of the diffusion potential, that is the ion mobility, ionic permeability, and the ionic concentration differences. Next is action potential. Basically, action potential is a change in the electrical potential associated with the passage of an impulse along the membrane of a muscle cell or the nerve cell. If we talk about the ionic mechanism in the action potential, then we will discuss about the types of ion channels and the properties of ionic channel. There are the three types of the ion channels: number 1 sodium channel, number 2 potassium channel, and number 3 calcium channel. These three channels play important role in the various functions. Sodium channels plays very important role in the action potential initiation and the propagation in the excitable cells, including nerve, muscle, and the neuroendocrine cell. And if we talk about the potassium channels, these play important role in the action potential, like returning the depolarized cell to a resting state. And third is the calcium channel. These channels play important role in the many body functions such as the cardiac action potential and muscle contraction. Last is the properties of the ionic channels, in which if we discuss about the threshold is any action potential, any stimulus that is initiate the action potential. Action potential required stimulus to start, to initiate, which is known as a threshold. And if we see all or none response, so it is a response of the any stimulus that is present either or not. If the stimulus is present to initiate the action potential, it is known as the all response, and if it is not present to initiate the action potential, it is known as the non-response, all or non-response. Next is the latency. Latency is the time period between the onset of the stimulus current and the peak of the ensuring the action potential. Next is the strength duration relationship. These both are very important to initiate for any action potential. And if we see refractive period is a period when a membrane cannot be stimulated to support an action potential. So that is about the types of ion channels and the properties of the ionic channels. Next is axonal propagation. Basically, spread of an action potential along a nerve cell will illustrate the mechanism of propagation, and you can see this diagram. First, action potential is initiated at the axon hillock, where the axon extends from the cell soma, and these action potential propagate along the length of the axon until it reaches the terminal synaptic. This action potential traveling along the nerve cell only in the one direction, and this direction is known as the orthodromic direction. Orthodromic direction means the correct directions. And next, this diagram. In this diagram, you can see first Sachuen cells. These Sachuen cells surrounds axons to form the insulation and play important role to form the insulation. And next is the nodes of Ranvier. And nodes of Ranvier are the thick lipid sheath of the myelin, which is interrupted by the gaps between adjacent to the Sachuen sheath cells. Myelination of the axons is an alternating means of increasing the conduction velocity. Now, I will tell you about the synaptic transmission. Synaptic transmission is the biological process by which a neuron communicates with the target cell across a synapse. Basically, synaptic transmission is a process in which specific information, transfer from the presynaptic cell toward the postsynaptic cell. For this, there are the two types of the transmission processes that are being used. First one is electrical synapse and the second is a chemical synapse. Here you can see electrical synapse and the chemical synapse. If we talk about the electrical synapse, you can see that there is the information that is transferred from the presynaptic cell toward the postsynaptic cell in the form of the ions, and there is a gap junction between the pre and the postsynaptic cell. This gap junction contains the connexons. Connexons basically helps the flow of ion easily from the pre toward the postsynaptic cell. And if we talk about the chemical synapse, there is the specific neuromuscular junction between the pre and the postsynaptic cell. And first neurotransmitter released by the synaptic vesicles, and the information transferred through this neurotransmitter from the pre toward the postsynaptic cell. That is about the electrical synapse and the chemical synapse. Next, this small video will describe your exact process or exact phenomena of the electrical synapse or the chemical synapse. If you see the electrical synapse, you can see that there is a gap junction between the pre and the post. And through this gap junction, information transfer from the pre to the post. And if you see the chemical synapse, you can see that the neurotransmitter first released from the synaptic vesicles through the specific neuromuscular junction toward the postsynaptic cell. And that is about the process of the electrical synapse and the chemical synapse. Now I will tell you about the neurotransmitters. Neurotransmitters are the chemical messengers that transmit a message from a nerve cell across the synapse to a target cell. The target can be another nerve cell or a muscle cell or a gland cell. They are the chemicals made by the nerve cell specifically to transmit a message. If we talk about the categories of the neurotransmitters, there are the four categories of the neurotransmitters. Number 1, acetylcholine. Number 2, biogenic amines. Number 3, amino acids. And number 4, peptides. Next, there are the different types of the neurotransmitters and their function. You can see like adrenaline. It is a fight or the flight neurotransmitter. Noradrenaline, it is a concentration neurotransmitter. Dopamine, serotonin, GABA, acetylcholine, glutamate, and endorphins. These all neurotransmitters are mentioned with their functions. Next, I will tell you about the neural integration. Neural integration is a way of opening up the communication pathways in the brain that may have become blocked or shut down because of stress. The practitioner can not only discover which pathways in the brain are not functioning to their fullest, but can also open them up and allow them to be available to be used. In which we will talk about the excitatory postsynaptic potential and inhibitory pre-postsynaptic potential. First is synaptic inhibition. If we talk about the synaptic inhibition, the process of inhibition is occurring in the presynaptic cell as well as in the postsynaptic cell. Basically, the process of presynaptic inhibition is a reduces the amount of neurotransmitter that is released by the excited neuron and causing the inhibition of the postsynaptic neuron. And if we talk about the postsynaptic inhibition, it works in the opposite direction, in the opposite that causing the inhibition of the presynaptic neuron. Basically, the function and the basic process of the inhibition is to inhibit the action potential to generate. Second one is the synaptic excitation. The process of synaptic excitation only occurs in the postsynaptic cell, and that makes the postsynaptic neuron more likely to fire an action potential. It allows the action potential to generate. When an active presynaptic cell releases neurotransmitters into the synapse, some of them bind to receptors on the postsynaptic cell, and this is the process of the synaptic excitation. Next, I will tell you about the facilitation. Facilitation is the enhancement of the response of a neuron to a stimulus following prior stimulation, and the response is dependent on the frequency of the stimulation and on the neuromuscular facilitation. You can see the diagram in which the response of a neuron is enhanced to a stimulus following prior stimulation. That is the process of facilitation. Last topic is the post-tetanic depression, PTD. They describe the reduction in the sensitivity of the synapse response, excitatory or inhibitory. to incoming the signals following a tetanus. This is generally caused by a reduction of the amount of the neurotransmitters on the hand at the synaptic terminal. When there is a reduction of any neurotransmitter, it causes a disease, tetanus. It causes the tetanus, which is also known as the post-tetanic depression. In this figure, you can see that is caused by a large accumulation of the calcium in the terminal caused by a high-frequency tetanic stimulation. That is about my presentation. Thank you so much.